Okay, friends, here we are, another podcast. I know you're so excited. Um, I need to talk with you today about some of one of the four major groups of organic molecules that we are just starting to learn about. These complex molecules based on carbon and hydrogen, of which all living things are actually composed. It's not that water and other inorganic compounds don't play a very important role in our living systems, but what actually builds our bodies are organic compounds. And there's four groups that we've mentioned. The first of these that we will study in detail is carbohydrates. Okay. Um, all right. When we think carbohydrates, we should think of all these things that you see on the screen before you. Sorry. Sugars, starches, things that probably are not so yummy, like this tree trunk we're looking at here. That's a whole lot of carbohydrate too. So all of these things are examples of carbohydrates. Um, Now, basically what you can help you remember what these are made of. Carbo referring to carbon, hydra referring to hydrogen. The 8 is always an indicator of an oxygen. So carbohydrate indicates that these molecules are made exclusively of carbon, hydrogens, and oxygen in varying amounts. But it's always in this particular ratio. It's always in this 1 carbon to 2 hydrogens to 1 oxygen ratio. Notice there's no nitrogen in this, no phosphorus in this. We talked about nutrient cycles. The one cycle that's really important for making carbohydrates is the carbon cycle, which is what directly involves photosynthesis and cell respiration. Just something to keep in mind. Okay, the most famous carbohydrate is this one. We'll talk quite a bit about it. That's the formula for glucose. It can be the formula for other things as well, but when we see it in here in our class, it'll generally be glucose. So sugars, starches, cellulose. Now, the functions of carbohydrates are as follows. It is a source for quick, immediate energy. It is also a way in which organisms can chemically store energy in their bodies for use later on. And it also has structural uses, meaning that it can actually be used to build bodies. It is generally not used to build animal bodies. Proteins are usually used for bodies like ours, but for a plant, carbohydrate is the primary building material. So energy and certain kinds of building materials. Those are the two main functions. Okay, quick energy and stored energy and building material. All right, the basic structure of a carbohydrate. First thing we need to do is remember what we learned about basic structure of most organic molecules, and it's this theme, this idea of a monomer plus a monomer plus a monomer gives us a polymer. So small building block plus small building block plus small building block is going to give us a big long compound. Okay, so if we take that theme and translate it to carbohydrates, what we end up with first, the monomers. What are the little basic building blocks that we're going to be fusing together to make more complex carbohydrates? The basic carbohydrate building block is called a monosaccharide. Saccharide always means sugar. So mono, of course, means one, so 1 sugar. Okay, that's your basic building block. Now, of course, we've talked about how carbon is at the center of all this. These sugars, sorry, somebody's playing with my lighting. These sugars are built of carbons and hydrogens and all that, but to the smallest thing that you can have and have it be a carbohydrate is a sugar. A carbon by itself is not a carbohydrate. You've got to you have to put them, the carbons and the hydrogens and the oxygens together to get yourself a sugar. Okay, so monosaccharides are known as simple sugars. Okay, they're also an example of a carbohydrate is glucose. Okay, it is a simple sugar. It is a monosaccharide. So these two terms are the more broad terms. This is a specific example. There are others. Okay. Now that's showing you a glucose molecule. That is showing you a glucose molecule. And what this whole diagram down here is showing, that if you fuse them together, you're going to get something else. But for right now, let's just focus on these two individual glucose molecules. Okay. Now, the word polymer, we remember that one. Poly means many. So a polymer, there you go. When we fuse together right here by removing water and doing a dehydration reaction, if you'll remember, we are going to create a covalent bond here, and that's going to create, well, yeah, and this is really not a polymer, because we just said polymer over here means many, and that too is not really qualify as many, but we're beginning to build a polymer here. This particular covalent bond responsible for linking two sugars is given a special name. It is called a glycosidic linkage. It is nothing but except a fancy name for a covalent bond between two sugars. That's it. That's all you need to understand. Okay? So, and we've just been over that, glycosidic linkage. Okay? All right, basic structure of a carbohydrate polymer then is a whole bunch of sugar, a whole bunch of them linked together in a long chain. Monomer is going to give us a polymer. Okay, so this whole big long molecule is a polymer, of a carbohydrate polymer made of many sugar monomers. Okay, and each of those pairs of sugar monomers, or each sugar is linked to another one by those glycosidic linkages or covalent bonds. Okay, that is a recurring theme. We will take these out and replace them with other things when we're talking about proteins, for example. So we'll see this monomer to polymer theme with the covalent bonds in other groups as well. Okay. All right, some basic information about sugars. Just this will be of help when you're taking a test or anything else. Basically, you should remember that OS, if you see OSE as a word ending, we're probably talking about a sugar. Usually they're classified according to how many carbons they have. For example, glucose has 6, pentose sugars, hexose sugars, of which glucose is an example, have 6, hence the 6 carbons is a hex. You won't need to know that. 5 carbon sugars are fairly common. We'll use ribose a fair amount of times. I wouldn't worry about these terms right here. They're useful, but they aren't very used very often. Glucose and ribose, though, we might use relatively often. There's 3 carbon sugars, so these are much smaller, more simple than these that take on these ring structures. That's a glyceraldehyde. Just so you know, we'll use that one as well. You don't need to knock yourself out on these. This is just showing you how these functional groups, this carbonyl associated right here versus a carbonyl associated in the middle of the sugar molecule somewhere, one on the end versus one on the beginning, kind of changes the personality of the sugar to some degree. These are all sugars. They're showing you a particular way of diagramming them. I wouldn't sweat this diagram very much. I'm going to skip it right now. All right. I do want you to understand this diagram. This is just showing the different ways that exist to notate or draw a sugar molecule. That's it. That's all it is. All of these are the same sugar molecule drawn in different ways. This is how we draw a sugar molecule linearly. Notice it's carbon chained to carbon, covalent, these are all covalent bonds between carbon, 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 and the carbon is covalently bound to some hydrogens, to hydroxyl group, this group up here. So that's one way of showing this particular sugar. The other way is to show it how it more realistically exists in nature. Most sugars that are this many carbons, 6 carbon sugars, don't exist in a linear form in nature. In aqueous solution, which is where you find most things in nature, in water, in living things, they take a ring form. The bonds bend and they take this ring form. And in reality, the way you will often see these ring structures made is like this one over here. What each bend in the ring, you'll notice the carbons are shown here, 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 and here, but they're not shown here. It is assumed that you will understand that anytime you see a bend in a carbon ring, you will assume there is a carbon there, okay? So you have to understand that that's carbon. You should recognize this ringed carbon structure with H's and OH's hanging off of it is going to be a sugar, and 9 times out of 10 it's going to be a glucose. You should be able to recognize that molecule, and you need to kind of understand what it's showing you. It's showing you these carbons covalently bound to each other to form a ring and to hydrogens and hydroxides, okay? And I've lost my thing. Hold on. All right. All right. And we know that we find lots of water in cells. So now, if sugars are in fact energy molecules, as we have mentioned, then where is the energy in these molecules? How in the world do they store energy? The energy in energy storage molecules, sugar, starch, even in fats that we'll learn about later on, they aren't carbohydrates, but is always in the carbon-hydrogen bonds, the bonds linking a carbon to a hydrogen. Why is that? Okay, well, we will get into this some additionally later, but a carbon and a hydrogen share their electrons very, very equally, okay? And so if we, as opposed to, for example, an oxygen and a hydrogen, whereas the electrons, because of oxygen's electronegativity, they're going to be much more towards the oxygen. Since that is the case, they are already, when they are near an oxygen, there's not much energy left. They have gotten to their lowest energy state. They are being hugged so tightly by oxygen. It's like you've dropped the book on the floor already. It's at a low energy state. You're not going to be able to get any more energy out of that book. You've already pushed it on the table, off the table, and it's on the floor. But here, this is though the book is still on the table, and if you break this bond, these can move to lower energy levels when they attach to the hydrogens attached to other atoms and the carbons attached to other atoms, and so that releases chemical energy. Bottom line is, because these share electrons so nicely, they have a lot of energy to give when we break this bond. Okay, so you can see that there's a lot of carbon-hydrogen bonds in this molecule. Here's one, and here's one, and here's one, and here's one, and here's two up here. You just can't see them drawn separately. So there's a lot of energy to be had if your body is able to break those bonds. And fortunately, our bodies are, and the bodies of almost all living things, are designed to break those bonds really well. So we'll talk about that some more. There's all your carbon-hydrogen bonds. Okay. Now, there are different types of sugars. There's simple and complex. Of course, monosaccharides, one sugar building block, are simple sugars. Glucose is a great example. One carbon ring molecule, that's it. Disaccharides are made of two sugars stuck together. Okay, an example of that is this maltose. We see it here in this container of malted milk. That's full of maltose, milk, disaccharide sugar. Polysaccharides are larger polymers, of course. Polysaccharide, polymer, poly means many. So they are many sugars linked together. A great example is starch, and we'll talk all about that. Okay, so monosaccharides, the main function, we're going to kind of go through our different sugar types in some detail. Main function of a monosaccharide is to give you quick energy. Okay, glucose is the best example for that. That is the form of sugar. Glucose is the form of sugar that is most readily usable by human or any living thing's cells. It's the fundamental molecule that enters into the cell respiration process. You remember we've beat to death that photosynthesis makes glucose, cell respiration breaks it down. It is the fundamental molecule that enters into that process of breaking down food to get energy. There are other monosaccharides. One of them is fructose, one of them is galactose. Fructose is found in fruit. Let me just jot that down. It makes sense. It is the sugar found in fruit. Galactose is the sugar that is found in milk, or one of the sugars found in milk, and we'll talk more about that later on. So, when we build more complex sugars like disaccharides, we do a dehydration synthesis, which means this is going to lose an H, this sugar is going to lose an OH. They are going to come together forming water. That's going to leave, and what's going to be left behind is this covalent bond called a glycosidic linkage. Okay, that is no big fancy thing. That is just the name for the specific type of covalent bond. Okay, glucose plus glucose gives you maltose, the sugar that is in malted. If you have ever had a malted, or if you like malted milk balls, you know, and milk eggs at Easter time, that's what you like. Okay, is maltose, that flavor of that particular sugar. Okay, now if you take glucose and fructose together, if you're going to build a different disaccharide from maltose, what you're going to build instead is table sugar. So this is taking glucose, that standard sugar that is readily used by all living things in cell respiration. You're going to do a dehydration synthesis with fructose, and that is going to give you common table sugar. Okay, glucose plus fructose gives you regular table sugar that is you sprinkle on your cereal, is in your candy, whatever. Okay, now there's another important disaccharide called lactose. Lactose is the true milk sugar, but it is made of two, again, it's a disaccharide, so it's made of two monosaccharides. It is made of galactose and glucose together with a glycosidic linkage in between them, and that gives you lactose sugar found in milk. Okay, now what is the point of disaccharides? We know that monosaccharides like glucose give us quick energy. Disaccharides give us energy, but not quite as quickly. And the reason it's not quite as quick as a simple sugar is because before this can enter into cell respiration, we've got to break an extra bond. Okay, now that is useful for organisms, because if all we had in our bodies were those simple sugars, they would be used up very, very quickly in cell respiration or whatever. But this disaccharide allows sugar to be moved around or kept in an organism for a while without being necessarily used by that organism. Let's think in terms of plants. Glucose, of course, is made in the leaves, but it must be stored in roots. That's where plants store a lot of their nutrients, is in their roots. So glucose made up here by photosynthesis has to somehow make it all the way down the tree into the roots without being consumed on the way down by the plant. Okay? So, what nature has done, what natural selection has done, is that it has favored the joining of that glucose and fructose to make sucrose, a disaccharide. And sucrose, that disaccharide, is able to make its way down the plant very effectively and into storage in the root without being broken down so readily because of that bond that exists between those two monomers, the glucose and the fructose. It's a little harder for the tree to break that bond, and so it survives its journey on down to being stored. Okay, and that's just showing you once again the sucrose table sugar. Okay, so prevents glucose from being so easily consumed as it moves its way down the plant. Lactose is a similar thing in mammals. That's just a disturbing picture. But in milk, in mammalian milk, okay, cows produce highly nutritious milk for their little baby calves, but this milk would be highly nutritious for anything. It would be highly nutritious for the adults as well. And this could be a problem if this was still nutritious for adults. You might have adult cattle trying to get this resource that the young really need in order to help them grow. The lactose solves this problem. Remember that glucose and galactose bound covalently here by that very important little bond, make up the lactose sugar. The thing is, a special enzyme is required to break this bond, okay? And only young mammals have it. As mammals get older, they don't have it anymore. In fact, if you know of someone who is lactose intolerant, it is because they lack the enzyme to digest lactose. And that was the normal state for mammals, including humans, except for in reasonably recent times. The vast majority of the world's population human population is still lactose intolerant. I don't remember if I have the map in this PowerPoint or not, but it's very select areas that are made up of mostly lactose people who can digest lactose. It's a racial, breaks out along racial lines. It's Asians, I believe, are highly prone to not being able to digest lactose, and that is the normal state for mammals. So that means, the bottom line is what that means is if you're an adult cow, you could try to drink this, but you're going to get indigestion when you do because you can't break it down and your body doesn't handle it well. So that means the adult cattle leave the mother cows alone, and the resource goes to the only organisms that can actually break down the lactose, and that's the babies. So we eliminate that competition for the needed resource. Okay? Polysaccharides function is energy storage. Energy storage, one function. And that's long-term storage, not this kind of temporary storage or this protective maneuver that we've just learned about. Long-term energy storage in plants. If you have ever eaten carrots, you are eating food that the carrot plant, which is the main green part up here, stored in its root for later use when times were tough or whatever. Same thing with the potato, although that's not the root, that's actually a funked out stem, but whatever. There's another different form of carbohydrate storage form in animals called glycogen. It looks a lot like starch, but it's not starch, and animals cannot make starch, so they use a different chemical for that. And glycogen is generally found in your liver and in your muscles. Now, the structure of, I'm sorry, this is talking about not the molecular structure, but the use of a polysaccharide as a structural compound. Cellulose in plants. Hopefully you will remember that cellulose builds cell walls in plants, and it is the main structural component of plants. It's what holds plants upright. A tree trunk is built of cellulose. It's what makes it so durable. So, these we easily digest. We make them in our own bodies to be consumed later, or we go and we, big bullies that we are, steal the potatoes and the carrots and we eat them and we digest them. This stuff, on the other hand, is not digestible, and we, generally speaking, do not go and break off big hunks of bark when we get hungry and eat it, because it's not, we can't get the nutrition out of it. We lack the enzymes it would take to break down that cellulose. Even though it's a polysaccharide, just like these are polysaccharides, it's different enough that we can't break it down. Chitin is another structural carbohydrate, and it builds structures in these bugs and in these crustaceans that make their exoskeletons hard and able to protect their internal organs. There is also a lot of chitin in the cell walls of this Gretabiga mushroom. It is the cell walls of that are made entirely of chitin. Same stuff in this bug, so when you step on a bug and hear it go crunch, you know, what you're hearing is the same stuff you eat when you are eating a slice of pizza with some mushrooms on it. Okay? All right. Polysaccharides can be very boring and linear. Okay? This is and I know it's drawn like a little wavy thing, but essentially this is a straight line of sugar molecules making up starch. This is glucose, 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 glucose. Do I really have to go on and name all these glucoses? Because I will, but that's glucose. A bunch of those glucoses linked together is called starch. More properly, chemically, amylose is the proper name of starch. There's another type, amylopectin. I wouldn't sweat it. Now, let's look at glycogen. How is glycogen different from starch? Well, let's look at it. I'm telling you it ain't but so different, because look, glucose, 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 all these glucose, 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 blah, 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 blah. How is it different then? Well, it has many more branching points. On starch, you don't see anything branching off from here. This is a straight shot, straight line. These have many, many branching points. That is how glycogen is different. We cannot make this, but we can, as animals, make glycogen in our liver and in our muscle cells. Okay, and if you think about it, this makes a lot of sense because muscles need a lot of energy. This is a source of piles of glucose, so it's a source of lots of energy. And the liver is just another place where we can store some of this stuff. It's probably important that you understand that you only have, I think I've got this right, about a 24-hours worth of glycogen stored in your body. So if you do not eat for a 24-hour period, during that 24-hour period, you will be drawing off your glycogen stores for energy. And it's only after that 24-hour period that your body would actually shift over to burning fat. Okay? So, starch made by plants, glycogen made by animals. Both of them are for storing energy long term. Now, this is a difference. This is a slow release compound. This is a pretty quick release if you need energy. For example, if you eat your breakfast this morning and you have to wait till you get lunch later on, you are not going to fall unconscious, okay? when you run out of your orange juice and toast that you consume this morning, when your body, you will begin, your body will start to just chip off bits of glycogen right, whoops, there we go, here, here as you need them, so that you don't just fall out and faint until you eat again. So this is a pretty fast release type of maneuver, where with a plant it's going to be a slower, and that's what the branching does. I'm sorry, I'm getting ahead of myself. The branching here provides many points where enzymes can start to work on this molecule to break it apart, whereas here you've only got this point where the enzyme can work and this point where the enzyme can work. So the branching makes this thing such that you can break it down much more quickly, and that's why there's the branching involved. Okay? I don't think we need to worry about this. Let's look at a better slide. You can digest starch. You have an enzyme that is designed to break this glycosidic linkage, this covalent bond right here. And you do it all the time. When you eat bread, when you eat potatoes, whatever. Okay? However, cellulose is not digestible. And if you look at this molecule, cellulose, this long thing here, is almost identical to starch, that long thing there. Okay? They really are almost identical. The only thing that differs is the nature of this covalent bond between them. And if you'll notice, they've got this kind of zigzaggy thing going. And we do not possess an enzyme. Someday I'm going to figure out why this does this to me. We don't have an enzyme that can interact. Look, did you see that poor enzyme go away? Nobody to play with. He can't play with that. It doesn't work. Okay, we don't have an enzyme to break that down, so that would remain in your body intact. Okay, if you ate some tree bark, it's going to come out pretty much unchanged because you don't have enzymes to break it down. Okay, so. Not everything lacks the ability to break down cellulose, though. Okay? Who can live off this stuff? Well, interestingly, bacteria, certain kinds of bacteria, can live off this stuff. Herbivores have generally adapted to have a certain type of bacteria in their gut. Herbivores like cows that are eating grass all the time have to be able to break down that cellulose and get energy out of it. Bacteria in their gut are what allow them to do this. Most carnivores don't have a relationship like this with bacteria, so they eat meat and so on to get much of their energy and nutrients. Okay, cellulose for us is undigestible roughage, which may clean out your digestive tract for you, but it doesn't give you much nutrition. Okay, these guys, on the other hand, are really able to digest those cellulose walls, as are these gorillas, and they don't need to ingest any other sugars. Okay. These guys, I'm sorry, are more like us, okay, in that they can't really digest that cellulose cell wall either, which is why they will eat fruit. But the cows can digest it, and here's why. They have a much more complex digestive tract than we do. I want you to look at this. This cow has, let me make sure I'm tracing this out properly, 1, 2, and then we're going to send the stuff up to get chewed again, chewing my cud, that chewed up grass. We're going to chew it one more time, and then we're going to send it into here, this chamber, and then whoo, through here, and then into the intestines. Would you please look at the length of that digestive tract and the fact that the poor animal has to chew it twice, okay? But this animal is adapted to break down this very difficult to break down cellulose, and it's got bacteria living in their digestive tract that actually are going to break down that cellulose for them. And so they kind of percolate this food in their stomachs for a long period of time, allowing this bacteria to work, okay? So herbivores tend to have have longer and more complex digestive systems than carnivores. Why? Because it takes so much more effort to break down that cellulose. Animal material is so much more easy to digest than plant material is because of those cellulose cell walls. It's very difficult. The digestive system to digest plant material has to be much more complex than what you or I have. Okay? These animals are called ruminants because they ruminate or chew on the food twice and get it really broken down. So let's go back. Let me go back. Let's compare these three polysaccharides. Remember, these are all polysaccharides. All right? Starch, glycogen, and cellulose. Starch looks like that powdery stuff. Plants make it, and plants use it to store energy. Animals can use it. That little girl is eating some nice starch, but she can't make it. We cannot make it. We can use it, but we can't make it, and we use it for energy purposes. Glycogen, on the other hand, animals make it, so we make that in our liver and in our muscles, and we use it for short-term energy storage, like you've got a day's supply of glycogen. Beyond that, you're going to have to rely on something else to provide you with energy. Cellulose, plants make it and use it to build their bodies. This beautiful tree is built of cellulose. Whoops. Animals cannot use it, okay? At least most animals can't, unless they have bacteria that are helping them out with that. Then they can use it, okay? All right, that's the end of the carbohydrate podcast. I hope that made sense. If not, come to class prepared with questions, and we will attack these things and make sure you understand it. We'll build some models and so on. All right, thanks a lot, and I'll see you soon. Nighty-night.